Cool. So yeah, today I will be telling you about in vitro and in situ iron geochemistry and microbial activity in Chocolate Pot's hot spring. To start with, I'll show you all where my field site is. Yellowstone, if you have not been there, first of all, I recommend you go there. But Yellowstone is in the northwest corner of the state of Wyoming. And my field site is just below the Norris Geyser Basin, about 5 kilometers south, along the Gibbon River and on the northern edge of the Yellowstone caldera. Here we have a face-on view of my hot spring. Indicated in blue is the flow path of the main hot spring. There are 2 satellite vents, but I did not study those in this line of research. And these 2 arrows indicate the sample sites from my master's research, one at the hot spring vent, one about midway down the flow path. And this is a little different from some of the other hot springs in Yellowstone, perhaps some of the more famous ones. This is a circumneutral pH hot spring, and I like to say it's more of a warm spring because the vent is about 50 degrees Celsius and cools off a little bit midway down the flow path. And like I said, it's circumneutral, very slightly acidic at the vent. This is a result of hydrothermal acid sulfate groundwaters mixing with alluvial groundwaters, and then you get the slightly acidic vent water. And up in the corner are just some of the properties of the water. There's a lot of ferrous iron in this water, near saturation with silica, and there's also an appreciable amount of sulfate, and this will come up a little bit later. So just go over briefly the initial study that I did for my master's. So this was more of a culture-based study. The main goal was to determine the potential for iron-reducing organisms in materials taken from these 2 sample sites. The first thing I did was a most probable number enumeration, and this is a technique to get an estimated number of the organisms in these communities. And following that, I did a series of enrichment cultures, and in these cultures, they were able to sustain their level of activity for more than 30 transfers, well over 400 days. And I also did some 16S and metagenomic microbial community analyses, and this identified well-known and potentially novel dissimilatory iron-reducing organisms. So here we have the activity for the cultures for, like I said, well over 30 transfers, well over 400 days. They just passed their 1000th day of existence a little while ago, so I'm really excited about that I've kept them alive and happy for this long. And like I said, there's this sustained level of activity, call it about 30 millimoles per liter of ferrous iron being produced. And if you're not familiar with metagenomics, don't be scared by this. This is the one of the metagenomic results from my vent community. This picture here, each one of the colors represents a different organism, and the shapes don't really mean anything, but they all cluster together, each of those dots clustering together. Those are all part of all of the DNA coming from one organism. So this is perhaps a more intuitive way of looking at it. These correspond. It's a way of teasing apart the number of organisms that you have in your metagenome. And from the vent metagenome, I identified 8 taxonomic groups, call it 8 different organisms that are dominating this community. 4 of these, I was able to recover nearly complete reconstructed genomes. So I have almost the entire complement of DNA from those organisms. And for 4 of those, I had hits for the PCC porin. What is this PCC porin? PCC stands for porin cytochrome complex. And this has recently been identified by Liang Shi from PNNL as a gene complex that has been found in all Geobacter species, which are well-known iron reducers, and also in some other species, one in particular, Ignavibacterium album. If you haven't heard of it, that's okay. But what is cool about that organism, while it has not been implicated as an iron reducer, This guy here, Melioribacter roseus, is a relative of it, a cousin of this Ignavibacterium. And I identified this organism in my enrichment cultures. And it also has this porine cytochrome complex, which is exciting because it has some implications that it's involved in the iron reduction cycle. Moving on to the current study. So the main goal here is to look at the in situ community. My previous study was looking at cultures. I want to know what's actually going on at the hot spring. So this is looking at the using the endogenous electron donor pool and again determining that iron reduction potential. And once again characterize the microbial community, so far looking only at 16S sequencing. Again, we have the hot spring. Here is the flow path again, and these symbols in red are spots that we took core samples. This was from 2 summers ago. 6 spots along the flow path. Here we have some pretty field shots. Here's the flow path from the top, one of the core samples we collected, and then this is the pool that forms at the top of the hot spring. So there's a couple different treatments for this experiment. There were, I added, there were treatments with and without additional electron donor, and treatments with and without molybdate. And the molybdate addition was to knock out any sulfate-reducing bacteria, which might be contributing to the amount of iron being reduced from sulfate reducer's byproduct, sulfide, which can chemically react with iron. Here's the results from that set. So this is site 1, that means the hot spring vent. Site 3 is the third core site, which is about 2 meters away. And the main takeaway from these figures is with and without additional electron donor and with and without molybdate, there's not a whole lot of difference at site 1. However, you move just the short distance away, the purple and green symbols are the ones that were amended with additional electron donor, and there's a much increased level of activity. But again, there is not much difference with and without molybdate, so the takeaway from that is there's not a significant influence from sulfate reducers. This is another measure of microbial activity. This is the measure of Fe2 over Fe total. The more Fe2 that's produced, the more active they are. So once again, you have a high level of activity at the vent, diminished when you move a little bit away, and at site 5, there's almost no activity. So what does this mean? We're still interested in where the electrons, or excuse me, where the carbon source is coming from. This is by no means a closed system. There is plenty of influx of other plant and animal debris flowing into the system, but you see this decrease in activity. And my thought is, if the only source of carbon is this exogenous material coming in, you would see a more similar level of, similar level of activity throughout the hot spring, but you don't see that. So to me, this would suggest that there is a source of carbon coming from the hot spring vent. And to support this, from the core samples that were collected from the vent, we identified relatives of siderooxidants, which is an autotrophic iron oxidizer. So there's good indication that this carbon might be being produced in situ. So the vent is the hotspot of activity. So comparing these different communities, this is a principal components analysis plot. Each one of these symbols represents an entire microbial community, and this is a way of very quickly looking at how related all of these communities are. So in blue, we have everything from site 1. Again, this is the hot spring vent. This is the T0 of my in vitro reduction assay. This is day 20. All of those treatments cluster very, very closely together. This would mean that there is not much difference between the microbial community. And what it also shows is that activity was stimulated by the addition of electron donor, but that the microbial community was not changed as a result of it. And I would also like to point out that within these communities, there is also that organism I mentioned earlier, Ignavibacterium. I did get hits for this organism in here. So again, this is all leading up to what is potentially the in situ iron reducer. So my latest line of research is a stable isotope probing experiment. This is using carbon-13 labeled acetate to probe the microbial community and, again, look for that those active organisms. So this looks fairly similar to the results from my in vitro reduction assay. The activity is stimulated by the addition of electron donor, but in green here, there is still an appreciable amount of activity without additional electron donor. And some conclusions. There's an active community at the vent. The community appears to be electron donor limited within a short distance of the vent. There are organisms related to dissimilatory iron reducers that I identified in my enrichment culture. And I did not have time to go into the iron isotope fractionation, but we are also looking at that to see if there is a biosignature of the microbial activity with distance and depth from the vent. And with that, thanks to everyone who funded me and organized this conference. And with that, I'll take questions.
Okay, we have time for a few quick questions.
Okay. Last month I was in Yellowstone and I saw warning signs all over hot springs. How acidic they could be and why they are so acidic that you cannot even touch them?
Yeah, so like I said, with my hot spring, it's a little anomalous from the rest in Yellowstone. But yeah, the extremely acidic ones, there's a lot of sulfide in the groundwaters. So I think that's probably sulfuric acid is probably the most of the source of acidity in the hot springs.
Anyone else? Okay, I guess we will move onward then. Let's give Nate one more round of applause.